Today I want to explore what creatures we have found living in the flooded quarry we are restoring, and also explain how this is going to help us decide on the next steps for this project. In total, we found more than 200 species living here in the various lakes, including some surprises and some odd creatures which I had never heard of. And if you have no clue what I'm talking about, then I suggest you watch the first 2 videos about this project. You'll find the playlist in the description down below. Okay, so to detect species in the project area, we're resorting to a mix of 2 methods: conventional surveying and eDNA. And for this video, we are only looking at the eDNA results. We purchased and conducted tests for both vertebrates and invertebrates to see what is living in 8 different locations in the quarry, which we labeled with these letters. This whole process started with the collection of the water samples from the various locations, which wasn't always easy. These samples were then passed through filters, which catch the bits of DNA in the water. It is really important to be careful not to contaminate the samples, and we mostly did a great job, only losing sample A due to contamination. All of this material is then sent to a company called Nature Metrics, who do the analysis.
And so, the process in a nutshell is extract the DNA, amplify the DNA that you want to know about, so fish or bacteria or vertebrates. And then once you've amplified the DNA, pass it through a sequencer. And once you've got all that wonderful data off the sequencer, pass it to your bioinformaticians, who then process the data and give you a list of species.
And by the way, this is accessible to the general public as well, in case you want to learn what lives in your area. So to interpret these results, a quick biology recap is in order. Let's use our little friend, the superstar frog from the last video, as an example. From lowest to highest taxonomic order, we describe an animal with species, genus, family, order, class, phylum, kingdom, and domain. And what this eDNA test aims to achieve is to get as close as species level as possible. In other words, the lowest taxonomic order possible. But when this is not possible, to give the nearest approximation. In our case, the test detected 31 vertebrate taxa and 173 invertebrate taxa. Among our vertebrates, of the 31 taxa they found, 22 can be linked to a species, which they name in their report. 4 of them can then be linked to genus level, 3 to family, 1 to order, and 1 to class. So we know that these 22 vertebrate species are there, and for the other stuff we detected, we have the best estimate of what might be there. Then, adding our invertebrate results on here, you can see that we have found a total of 204 taxa, and positively identified at least 63 species, and then have estimates for another 141 species. Okay, so with that biology lesson out of the way, what did we actually find? Let's start with the vertebrates before we get into the weird stuff. We found 6 mammals, namely red fox, beech marten, wood mouse, common pipistrelle, the endangered European rabbit, and of course the near-threatened Eurasian otter, which we spotted in this lake here in past visits. Then we found 16 bird taxa, of which 11 were identifiable at species level, including the common kingfisher, which I would really love to film here, the grey herons we see nesting in the main lake, and other wetland species such as the water rail, the common moorhen, the dunlin, and the Eurasian coot. We also found a random mix of small birds such as the dunnock, great tit, European robin, and song thrush. The remaining bits of DNA were harder to identify. We found thrush species, perching bird species, pigeon or dove species, duck, goose or swan species, as well as generally species of the Aves class, which, isn't particularly surprising. And I think at this point we can add a small caveat for eDNA, which is that when we find something, it is very likely that the species is there. But if we don't find it, it doesn't necessarily mean it isn't there. I say this because more than 118 species of bird have been spotted here by birders and surveyors, and they've been reported on eBird, which by the way is another useful tool for you to pick up and use in your area. So eDNA is more of a complementary tool rather than a silver bullet. But where this comes in really handy is when trying to figure out what is living in the water. We of course found the DNA of the European pond turtle, which we filmed in some of our new lakes. They are a near-threatened species and are the only reptile on our list. In terms of amphibians, we found 4 taxa: the near-threatened Iberian ribbed newt, the beautiful marbled newt, the Iberian water frog, otherwise known to Mossy Earth viewers as superstar frog, and some mystery toad species. Now, you might wonder what the point is with all of these lists. And, well, I thought it would be really fun just to show you what is living here, but the useful bit for us happens when we get to the list of fish species. You see, the main goal with this was to see which species of fish lived where, because we have invasives which can negatively affect other species. This is crucial information, because we are linking some lakes together, and we definitely do not want to help spread the invasive species. We found 4 species of fish, and now let's take a look at each one. The first one we have here is the common carp, which can grow to 80 centimeters in length. They search the bottom sediments for food and can feed on the submerged vegetation, which in turn can be detrimental for other species which depend on it, such as certain wetland birds. On this map you can see their native range in green, and where they have been introduced in orange. Then we have the eastern mosquito fish, a small American fish that can grow to about 6 centimeters and be detrimental to amphibians and many other aquatic vertebrates through their aggressive behavior. The next invasive species on the list is the largemouth bass, which is another American fish and an infamous invasive species which will predate on pretty much anything. It was probably introduced here by fishermen who wanted something interesting to catch, as it's a common angler species. Now, we knew of these 3 invasives, but using eDNA, we were actually able to find one which we were not aware of, the tench. It's another angler species, but probably not as damaging as the carp. If we then grab this list of species and match it with the samples in which they were found, we're able to lay this onto our map of ponds and lakes. As you can see, Area D was the only one where carps were found, which means that we should not connect it to any other bits, because they are the most damaging invasive. Once again, I must remind you that this data is incomplete. I saw some fish in Area H, but none showed up in the eDNA. Nevertheless, we want to be cautious, and so we are assuming that if carps were not detected in other lakes, it is because they're not there yet. So it is informing our work. Then the invertebrates report is rather overwhelming. I mean, some of these species are tiny, and we are not sure what to do with the information. I mean, Euricerus lamellatus, Anchathoscyclops americanus, or Nais elinguis? These might as well be aliens as far as I'm concerned. So I thought I would ask our newly created and soon-to-be AI overlord to see if we could find, a few common names for some of these species. As you can see, it found something at least. And don't worry, I know the AI is very prone to mistakes. This interpretation is just to be used in this video. As you can see, we have a nice assortment of beetles, such as the Mediterranean beetle, the diving beetle, and the fenestered mud beetle, as well as flies, fleas, and midges, of course. This is a wetland after all. Then we also found traces of the duck mussel, which I thought was pretty cool. And finally, our biggest invertebrate was one we knew was there already, because of all the holes. Procambarus clarki, the red swamp crayfish, or Louisiana crayfish. It's another widespread invasive that lives in our wetlands, and the damage it causes is generally due to the holes it makes. These results will also allow us to monitor for species presence over time, and act as a baseline for presence, but not abundance. I hope you found this visualization interesting. And finally, remember that you can support our rewilding projects by becoming a member at mossy.earth. We implement projects across a wide variety of ecosystems, and we employ our own team of biologists to run many of our own projects and to work with our partners in other locations. And between all of this work, we try to be as transparent and accessible as possible by making, these videos, but also being available to chat and answer your questions whenever you might have them, through Discord or through calls and emails. Wherever, we are there to answer your questions. And we also try to show you how we spend our money and the impact that we are having. So we're doing our best here, that's the point I'm trying to make. The membership starts at the price of your local frothy oat milk latte, so it's cheap to join, and once again, you can do so at mossy.earth. Until next time, cheers!